So far, we have learned about the structure of DNA, where it resides, how it makes a copy of itself, and even how your cells make a copy of a cell. And then, of course, we've also learned about how our cells make sex cells, which is important for genetic diversity and reproduction. So this next section is going to focus on what is called protein synthesis. And basically, the word synthesis means to make. So that means that the function of DNA is to make proteins. And if we stop and think again about what proteins do, they have a lot of different jobs. Some of the jobs of protein include things like giving you different characteristics like your eye color or your hair color. Also, proteins are also enzymes, and enzymes are going to help you do a lot of different things, including digesting your food. Also helps you with skin repair, building muscle. So a lot of things are connected to proteins. If you have a problem, it might actually be connected to the fact that maybe your body isn't able to make the proteins that it needs to. And that's actually where a lot of genetic disorders come from. We're going to learn about, or we've already learned also, about a disorder called cystic fibrosis, which is connected to a mistake in your DNA, which we've already talked about mutations, and that mistake is directly connected to the wrong protein being made. Let's start with a quick recap then. We already know DNA has many jobs, that it stores the information that makes the proteins. And like I was just talking about a second ago, there's all kinds of jobs that those proteins do. We've also already learned the structure of DNA. We've learned that there is a phosphate on the outside, a sugar on the outside, and then the middle section here are the nitrogenous bases. So a phosphate acid, a sugar, and a nitrogen base, those three pieces make up the smaller pieces of DNA, which are called nucleotides. And we've also already learned that adenine always matches with thymine, and C and G, guanine and cytosine, always match together. So we already know about DNA. The only thing is that in order to make our proteins, we actually need RNA to do this. And the reason it's called RNA instead of DNA is it is a different kind of sugar that's in it. So RNA is going to use ribose sugar. So RNA is called ribonucleic acid, whereas DNA is called deoxyribonucleic acid. So ribose is the sugar in RNA, that's why it's got an R, and deoxyribose is the sugar in DNA, that's why it has the D right here. Okay? A couple of other differences you need to know between RNA and DNA. DNA is double-stranded, so we've already studied how it is a double helix, and we've studied how it replicates, whereas RNA is actually only single-stranded. We've also learned that in DNA, A matches with T and G matches with C, but there's a little bit of a change. DNA has thymine, but RNA has uracil. So that means that A and U go together in RNA. There is no thymine. There is still G and C, okay? So you can see in the center here, this is what is the same between DNA and RNA. Still has the adenine, still has the cytosine, still has the guanine, okay? DNA is the master plan. This cannot leave the nucleus. RNA is what are the builders of the proteins. This takes the master plan and brings it out to the ribosomes to be the builders of the proteins. Make sure to make yourself a note. DNA cannot leave the nucleus, and so it has to make the RNA and send that out into the cytoplasm in order to build the protein. Since the DNA is stuck in the nucleus, there's a few different types of RNA that get made, and there's a few different jobs that they have. At the top of the slide, I did a little recap. RNA, remember, is single-stranded. The thymine is replaced by uracil, and these are out in the cytoplasm. The DNA is stuck in the nucleus. So, a couple of different messenger RNAs that you need to know, or different RNAs. The first one is mRNA. The M stands for messenger. And what that means is that it takes a copy of the instructions to the cytoplasm and to the ribosomes to actually make the protein. So this RNA has a really important job. The M stands for messenger, which means that since the DNA is stuck inside of the nucleus, what we do is we take a look at the code of the DNA, and from that code, we create or make messenger RNA so that that can then go out to the ribosomes. Another type of RNA is rRNA. The R in this case stands for ribosomal RNA. And that is because it is going to combine with the proteins in order to make ribosomes. So ribosomal combines with proteins to make ribosomes. So rRNA, really what it does is it makes the ribosomes so that then you can make your protein. Ribosomes are the makers of protein. tRNA is called transfer RNA, is what the T stands for, and it's going to carry the amino acids to the ribosome, hence the word transfer. They are the carriers of the amino acids. So we're going to see how all three of these work together. There are a ton of videos on YouTube to watch about protein synthesis. Here's one. I've posted a bunch on Schoology as well, so feel free to watch any of them. So this is a basic overview of what protein synthesis is. We start with DNA, and it is stuck in the nucleus. It cannot leave the nucleus. And so we get the first step. Step number 1 is going to be called transcription. And basically, the job of transcription is to make messenger RNA so that it can go out into the cytoplasm. So then out here in the cytoplasm, we get the next part of protein synthesis, which is called translation. So this is step 2 out in the cytoplasm. And so in translation, what we have is a ribosomal RNA attaches to that messenger RNA that was made in transcription, and then the transfer RNA brings in the pieces, and the pieces are little amino acids. And these little amino acid pieces all clip together in order to make a protein. Let's break down those 2 steps. So our first step we just said was transcription. This is going to happen in the nucleus. And the reason why it has to happen is that the DNA can't leave the nucleus. So it has to get converted into RNA. And particularly it's going to be messenger RNA, mRNA, because it's basically taking the message. Now, there's a few things that happen here. The T's are replaced by the U. Okay, so if in our nucleus we had a strand of DNA and one side of it looked like this, okay, the other side would be T, T, C, G, G, A. Now, if we're going to make our messenger RNA in transcription, we're just going to take one side as our template side or our guide. So I've outlined in red here, this is my template. This is my guide that your body is going to use in order to make that messenger RNA. So what it does then is it says, okay, I've got an A right here. My messenger RNA is going to have a U, not a T, because messenger RNA does not have T. And it's going to look at this one and say, okay, I've got an A, so I need to have another U over here. So it's providing my template. So G is going to have a C, then C, my messenger RNA will have a G, my messenger RNA with this C here will have a G, and then T matches with A. So it used this side of my DNA that I'm outlining in red here as a template in order to create my messenger RNA. So this piece right here now is a copy of the DNA, but it's slightly different because it's got the U's, but it's pretty much still a copy of the DNA. And this message can now go out into the cytoplasm for the next step. Here's an image of that process of transcription, that first step that is happening out in or inside the nucleus. So here we have our DNA on the left-hand side, and we're going to enlarge it to look bigger. So we have our RNA right here being made. That's our messenger RNA, and it is using the DNA here, which you can see is opened up, kind of like the way it opens up when it's making a copy in replication. So it's opened up, and we can see we've got a T, we've got a C, and then the RNA over here matches up, and it's like, oh, A goes with T, and G goes with C, and over here we've got A, and then of course we have our U. And that's because there is an A over here. And A matches with T. If it was DNA, we can see the other side of the DNA over here. But when we're making the messenger RNA, we've got to put a U in there instead. So there's our strand of messenger RNA that's going to be ready to go off into the cytoplasm. Then we get step number 2, which is translation. Translation is going to be happening in the cytoplasm. And so since the DNA is stuck in the nucleus, the messenger RNA goes out into the cytoplasm. And it's providing the code or the instructions for making the protein. And so what happens is, because we have that messenger RNA code, it tells what amino acid comes. And the amino acids join to make a protein. So here's my messenger RNA that we had just made during that first step of transcription. And so let's say the messenger RNA has this message on it. So there's two things that happen. We have a ribosomal RNA, an rRNA, comes in here and he says, oh, look at this. We have something we need to make. And so what happens then is a transfer RNA, which your body makes these, comes on over to here, and he reads this code, and he says, okay, I can read this because C matches with G, C matches with G, and U matches with A. And that's how they know which one to bring over. And so this particular tRNA is carrying a little amino acid right here. And then a different tRNA has to come over to match correctly with the GUA. And the tRNA that comes over is going to be the tRNA that has CAU on it. And so this tRNA comes on over and he brings an amino acid. And then this amino acid from the guy over here, he connects to this one. And then this tRNA, he can leave. So this tRNA, he leaves and he goes and he grabs another amino acid because this one is now connected to this one. And the whole thing continues all the way down the line until you get a whole bunch of amino acids connected to each other. And all those amino acids connected to each other make a protein. Step 2, like I said, it's a little bit more complicated. So what do you need to know? You need to know what happens in the cytoplasm. You need to know that the ribosome attaches to the mRNA. This is our ribosomal RNA. And that a tRNA, a transfer RNA, brings an amino acid. I showed you how they match up. The part on the tRNA is called an anticodon, and the part on the mRNA is called the codon. So that's where I had let's say we have CCC on the messenger RNA, and then let's say we've got AUG. This set of 3 here is called a codon. And on the previous slide, when I matched them up, when the tRNA brought in the piece, it would match up and say, oh, well, this is how I know what goes here. These are the pieces that fit together. And so this set of GGG here, that's called an anticodon. And it brings that amino acid piece. Okay? And then same over here. If I've got AUG, it says, oh, okay, that would be UAC. And that anticodon right there is going to match with it. My drawings aren't always the prettiest, so here's a nice image. Again, watching some of this happening on some animated videos, if you go on YouTube, might be helpful as well. But you can see here we've got our messenger RNA, just like we said. We have our ribosomal RNA, our ribosome, right here that attaches in the whole process to help. And then we have our transfer RNAs. Here's a tRNA that had just dropped off this amino acid. We can see previous amino acids are connected here. And so here's the transfer RNA, and you can see it says ACC. Well, if we look over here on our messenger RNA, A matches with U, C matches with G, and C matches with G. So that's how it makes sure to bring the correct amino acid. So the set of 3 letters right here, that's called a codon. Make sure you mark your notes. That's called a codon there. The set of 3 letters right here on our transfer RNA is called an anticodon. And so they match up with each other and they can make sure they bring in the right piece. So if we look at this next transfer RNA that says UUU, it matches up with AAA and brings this amino acid right here. And then the next transfer RNA has CUA and it matches with GAU and it brings this amino acid. And here comes the next transfer RNA to bring in the next piece. So a protein functions according to if it's got the correct amino acids in the correct order. Okay, so A, U, G, and C, the genetic code is basically sets of 3-letter words, codons. So if it says CCC, that's going to bring a particular amino acid to it. If it says UUU, that's going to bring a different amino acid. If it says CUC, that's going to bring a different amino acid. So these 3-letter words, these codons on the messenger RNA, give your body the information of, okay, what amino acid do I need to bring in order to make the right protein? We have actually figured out what all of the different codes bring, exactly what set of 3 letters bring what amino acids. So there are 64 possible amino acids. Okay, we read this from the inside out. This is called a codon wheel. So what we're looking at here, we're always going to look at the messenger RNA because the messenger RNA has the codons on it. That's why this is called a codon wheel. And so there are special letters that tell us that we have to start or stop. For example, I'm going to start in the middle here at UAA. If I had the code UAA, then I would be told to stop reading the protein, stop bringing amino acids, stop what I'm doing. Okay? So UAA tells us to stop. Also, UAG also tells us to stop. So there are certain special codons that tell to stop. There's also one over here, UGA. That also will tell the protein to stop being made. There is also a special start codon. And the special start codon is usually AUG. It brings the amino acid methionine, but that is the special code that tells the DNA or the RNA to start making the protein. So if I were to say GGG, which amino acid would be brought? I'm going to start at the middle, G, 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 and glycine would be brought. In the overall process of making a protein, this means that our general instructions are in the DNA. The DNA then has to be made into messenger RNA, and then from there we get the creation of a protein through that process of translation. Now, there can be mutations or a change in the genetic code. This could be a mistake in the original DNA. It could be a mistake that occurs when the messenger RNA is made. So there's a couple of different types of gene mutations. You could have a gene mutation that makes a change in just a single gene. So let's say that you have a gene to make the enzyme lactase, which is to digest milk. If there's a mutation in just that individual gene and you can't make the protein, then that's just one little problem, and maybe you're lactose intolerant. Now, in terms of chromosomal mutations, this could be a change in a whole chromosome. For example, a chromosomal mutation, you might have all of chromosome number 1 that you got from your dad, but the whole chromosome has an issue, like a whole piece of it broke off, or a whole piece of it duplicated itself extra. So there's a whole extra piece. That would be a chromosomal mutation. You are going to need to know the difference between those two. We're going to focus on gene mutations next. I'm going to talk about 3 different types of gene mutations: point mutations, substitutions, and insertions and deletions. A point mutation is exactly as it sounds. It's a mutation in just one or a few nucleotides, or just a point in the DNA. Point mutations will also include substitutions and deletions, and if this gene is changed, each daughter will also have a mutation. So here's a good example for you. Here we've got CTT. The messenger RNA should read GAA. In a point mutation, we have CAT, so you can see the T has been changed to an A, and now the messenger RNA says GUA. So that's a single nucleotide changing. So a substitution is an example of a point mutation. That's the one that we just saw here in the last slide. We just changed from one base to another base. And that usually only affects one amino acid. Sometimes it can have no effect, and a good example of a point mutation is sickle cell anemia. That is a point mutation where it is just a changed base to a different base. Insertions and deletions have a little bit more of an effect. Sometimes they're called a frame shift mutation, and this is where we're adding a whole base or deleting a whole base. Okay? These can have huge effects. For example, here we have a sentence that reads, "The fat cat sat." Now remember, codons are read in 3-letter words. So this is kind of how your DNA is read, in 3-letter words. Well, if we have one change where something is deleted, the T in this case gets deleted, the sentence no longer reads "the fat cat sat," it reads "hef act ats at." And so this is a good example of one deletion having a huge effect. Cystic fibrosis is an example of a point mutation that can have dire, just very serious effects on your health. The other kind of mutation that I had mentioned at the beginning is an entire chromosome, so changing the whole structure of the chromosome. And there are also 4 different kinds of chromosomal mutations: deletions, duplications, inversions, and translocations. Chromosomal mutations tend to have more serious effects. For example, trisomy 21 means you have a whole extra chromosome, which means that you've got Down syndrome. Prader-Willi syndrome, you have chromosome 15 is missing an entire area to it, and you have poor muscle growth, can end up with obesity, lots of compulsive behaviors to it. Turner syndrome, you're missing a part of the X chromosome, so only girls are going to have this. Means that there's going to be a failure of puberty, and usually there's heart defects. These are just a few of the many examples of chromosomal mutations. There is a lot of information available on the internet if you're looking to learn more about any particular chromosomal disorder. Now, the effect of mutations. It could be the cause of mistakes. Usually, there is about 1 in every 10 million of your bases where there's a problem. Mutations can also be caused by chemical or physical agents. We now know that even things like stress can have an effect on your DNA. So it's not just chemical things like smoke and pollutants, but even what you are exposed to, like stress, that affect your DNA. Now, there are mutations that can be helpful. One mutation actually makes your bone stronger, so that's an example of a helpful mutation. And there are also mutations that increase your resistance to HIV, we've found. So not all mutations are bad mutations. Sometimes a mutation also can have no effect, so we have to keep that in mind as well. That can also happen sometimes. And some mutations can cause cancers, sickle cell anemia, or just really change a large part of a protein that does major jobs in your body. And so that's when we have problems.